So dinner and 3 talks, and we start with lecture. So Marianne is going to talk about information theory.
Yes.
Photo also is today at 10:30. Very good. So right outside, after this lecture, we'll have a photo with all the participants. Right.
Okay, great. So what I wanted to do is to talk a little bit about essentially high-dimensional or approaches to high-dimensional data in gene regulation and development. And I wanted to do this. I don't know exactly what Arvind Rao is going to talk about in the afternoon. I assume it's something to do with image processing, but I was hoping that Archishman and at least my talks in the morning can sort of feed well into this sort of introduction that I wanted to give. So first I wanted to maybe just say what is or how can we sort of interpret data in gene regulation and development in this high-dimensional framework. And I would say in development, often at least this part of the framework is still, I think, at least partially accepted, namely that there are certain inputs that are processed by cells to regulate cell fates. And so now the question is, what are these inputs? And that, of course, depends on the exact example that you're looking at. So so I wanted to maybe sketch out 2 systems briefly. One system that I worked a lot is like sort of the fly, essentially, where these inputs are essentially transcription factors. So these are transcription factors that are deposited by the mother into the fly embryo, and then they sort of diffuse or regulate gene expression there. And then also, there's a series of sort of networks of transcription factors that I would say are then processed in the cellular nucleus by this gene regulatory architecture, where what I'm sketching here are different promoters with binding sites for these transcription factors. There are also enhancer regions on the DNA that can be further away from these promoters. So there's like a whole network, a whole architecture inside that processes these transcription factors that then gives rise to these different cell fates, which we can describe either in terms of gene regulatory outputs, or we say we want to number these cell fates directly. So that's the fly. There are, of course, the same input applies, of course, to sort of more complicated animals as well, for example, like mice, humans, mammals, where I guess these inputs, I would say, are now larger signaling inputs, like the signaling networks that are regulated, for example, by Wind, Notch, FGF, BMP, and many others. So these are external ligands, sometimes other factors that can essentially give rise to these different cell fates. Cell fates here are often classified with other markers, which typically mark different sort of cell fates in terms of what type of tissue they form later, so mesoderm, endoderm, ectoderm, and so on. And they are typically marked by, probably many of you will have seen this, markers that like socks and bra and others. So this is on the output. And the processing, I would say here, is done through complex signaling pathways that basically, so this was meant to be a nucleus, that basically, if you have the full cell, you get sort of, things that act externally, some sort of ligands, and there's some signaling network that involves many, many molecules, many proteins, eventually some nuclear transport, and then eventually, again, inside the nucleus, there's gene regulation. So I would say the fly is a more simpler example of what's happening on the level of the processing. But then, of course, in more complex animals, you have analogous ideas, essentially. And then the final thing that I wanted to remark on is in terms of like, where is this high-dimensional data, or where do you get it, is, of course, that in addition to experiments in animals, there are, of course, experiments either in cell culture, where you can take stem cells and culture them and image them, or in these organoid structures, where, I mean, just to give a few examples, I think many of these organoids are, for example, done in mouse. One example, I think, is somatogenesis, where, what happens is that basically these vertebrae form from essentially the tail of the mouse, and now people can essentially isolate these tail tissues and make, two-dimensional analogs where you can then see these waves going through the network. So that's in the lab of Ina Sonnen, I think Alexander Aulela and other people, Alexander van Oudenaarde and so on. And then, of course, there are the gut crypt organoids that a lot of people use to also study wind signaling.
Marianne?
So, yes.
Could you write maybe a little bit bigger?
Oh, okay, yes, sorry. Yes, I'll write bigger from now on. Okay, perfect. Thank you, Gautam. Okay, anyway, so I just wanted to say there's, in gene regulation and development, there's this idea that there are lots of different inputs, there's complicated processing, and then there's on the output, essentially cell fates. And so now I wanted to talk a little bit about the two types of approaches, at least in, or two types of the many approaches that people have used to understand this. And I think what Artishman is mostly going to talk about, hopefully today, is to describe this development in the framework of models. In the framework of models, what you would do is that you basically write these outputs. I'm going to use the variable O, but there are, of course, many. You write these outputs as some sort of function of different signaling inputs. I'm going to use these inputs. I'm going to use the variable S for this. So you have different signaling inputs. You have, of course, signaling inputs at a particular time. You could have delay differential equations. And that, of course, also depends on a lot of different parameters. So you have essentially a nonlinear dynamic models for your system. And then in the framework of the model, I think people do typically one of two things. So the first thing is that you can look at the steady state of the model. Or you can look at essentially the dynamics. And I think both things you'll hear more about in the rest of the conference. In the view of the steady state, I think what people typically do is that they look at the attractor and the fixed points and the limit cycles in these systems. So you essentially, look at good that you're interested. Okay, no, all good. So basically, what you would do is that you basically look at the fixed points or limit cycles or attractors of these systems in some sort of nonlinear landscape. And so then you get, as I said, maybe a landscape that depends on fixed points, a landscape that has some, bistability in terms of these variables that you're looking at. So in terms of these variables, maybe, and then you classify the cell fates depending on these stable states. So often there's an idea in development that basically once you get to a stable state, this is, the cell fate that you develop into. There's going to be probably, I expect, a little bit of pushback against this, the steady state view next week, where I think Aneta Koseska will speak, who I think really believes in this type of dynamics. And there is already a little bit of pushback in the sense that you could just say, in some animals, if you have these outputs essentially fluctuating over time, maybe, if nothing has happened over time, even though the fixed point might be something else, maybe once the cell, has reached a particular time point, a particular marker, like for example, in C. elegans, the end of a larval stage, sometimes these timings are a little bit more strictly coded, then maybe it doesn't even matter what fixed point you're going to. You're just going to look at the dynamics that was stable for a certain amount of time, or that was stable at a particular time point where you need to make a decision. So that's, of course, an alternative view, but still fine. And in that case, I think and what I wanted to get to in both cases is that essentially, I think to some extent, but Artishman will probably maybe describe this better, is that in both cases, what you essentially look at is some sort of nonlinear dynamic landscape that you analyze. In this case, you analyze this landscape in the terms of fixed points, and then can do really nice things with respect to these fixed points, where I guess sort of Artishman will talk later, and then there's also sort of lots of different papers from the Sidhartha family, and I was told to write larger, so I'm gonna just write this larger. From the Sidhartha family that you can look at to sort of analyze, the type of fixed point structure geometrically in this landscape. And then on the dynamic side, what people were doing is that they are thinking essentially about this type of fixed point landscape and how maybe different environmental variables, or how it changes, how different environmental variables might change this landscape. Maybe new fixed points emerge, maybe new fixed points lose their stability, or how this happens as you change the environment, so that you basically have a complex temporally varying environment of stimuli, T. This often happens in sort of mouse development, where people say that you often have oscillating inputs, and that could then maybe permanently change your landscape. And I think what Aneta Koseska is going to talk about next week is essentially how some of these changes change the fixed point structures, and then these things emerge that are called ghosts of a fixed point, where essentially you no longer see the fixed point in your system, but you know, because a landscape that would have had a very similar parameter would have had a fixed point, your dynamics around this no longer fixed point is essentially slowed down. I would say that's sort of the model side of things. And then I guess where I want to go to is towards the optimization side of things, where basically what I would say is that you now no longer have the intuition that a particular model, that a particular type of differential equation describe your system, but you have the intuition that something in the system is optimized. And we have, of course, heard sort of, you know, in various parts of these earlier talks that, you know, maybe what is optimized is not always intuitive. But I think in some of these early developmental systems, there is maybe an argument that some amount of information flow needs to happen because you do have these inputs, you do need to develop into some cell fates. And I think it's good to actually really push back on the optimization, but it's also, I think, somewhat plausible that in early development, some of this is happening. Still, before I talk all the way about optimization, I just wanted to mention, to give a sort of semi-complete overview of how people or what one can do in these systems, is that I think an in-between sort of way is to just do max-end optimization, which is an optimization approach. But basically, in these max-end approaches, you still have, to some extent, an idea of what the model should be, what your model of the system should be, but now you're optimizing these parameters with the view that these parameters should be chosen such that your observables essentially satisfy maximum entropy principle. So you're putting a little bit, I would say, a little bit of optimization weight into your intuition, but you're also still having an idea of what the structure of the system should be like in terms of the models or in terms of, you know, the shapes that you might go up to second order terms or, you know, some approaches like this. And then also maybe for people who are interested in this, I feel like when people do these max entropy optimization, then I think a quantity that people often like to look at is essentially the Fisher information. And the Fisher information is basically a change of this function with respect to the parameters theta. And the Fisher information is also often looked at to essentially find, because what you can say that maximum entropy is doing is essentially, or that what you want to do is essentially finding unbiased estimates of your parameters. And the Fisher information can be expressed as a bound essentially that can give you a sense of how unbiased your estimate really is. So just to give you a view, and I'm also mentioning this because recently in terms of references, there's been a lot of work on maximum entropy approaches from Chris Lin et al. And then pseudo-maximum entropy approaches from Aurel et al. And I think there's a nice review maybe. I think this is misspelled. From 2017, if people are interested in this. Okay, so this is the maximum entropy approach with respect to the optimization. And now I wanted to say, you know, there's also model-free, or to some extent model-free optimization, which is where what I would call these information approaches, where what you now could say, well, you know, what if I look at these systems and I don't fully know what my inputs are or what the exact number of cell fates are, I could assume sort of that what I think the system should be doing is that it maximizes the information between these inputs and the outputs. And then you typically want to maximize over something. And often, I think in these systems, you have the strong intuition that on the output side, there are a certain number of cell fates. I know there are arguments about that, but basically if you have the intuition that on the output side there should be a certain number of cell fates, what you would optimize over is essentially the input distribution. So that's a standard optimization that people do, and that I will sketch out for one simple system on the board so that you know how it's done. But just to say, so this is then a probabilistic approach, and what you will mostly get is a probability distribution of these signals, which we will, and hopefully I will make an argument that you can maybe also do this to essentially find models. And then the other approach, the other standard optimization goal, I would say, also because I'm biased because I work on it, is essentially the information bottleneck approach, where what you're trying to do is essentially find this map between the processing variables and your inputs S. So you call now all your processing variables. I always use this variable C because this approach is practically a compression. So you have these processing variables C, and what you're optimizing, what you're trying to find is the conditional distribution between C given S. Actually, like when I write these distributions, people know from Shaun's talk what things mean. So PC given S, yes, great. So this is essentially the map that maps your processing variables from your signals, what your processing variables are doing. And you try to find this map by essentially saying, well, these processing variables should maintain as much information as possible about the outputs, because that's the thing that you care about. You want to get to the output. You want to get as precisely as possible to the output. But it does so for a constraint between the information between C and S. So on a constraint between the variables and the signaling inputs. And I realized I haven't written down the definition of the mutual information. So the mutual information between two variables can be written essentially as an integral over the joint distribution divided by the two marginals. And what it practically does is that it tells you how much you know about one variable when you know the other variable, essentially. So you can almost think of it like a correlation, but it's essentially thought of like a more complete object that essentially includes all these possible correlations in the system. Yes? Yes. Exactly, because it's so yes, basically because what you're saying is that here, and this is a very good point. So what you're saying is that basically you're assuming you have tons of input signals on some level. Then you have this intermediate variable, and then you have the output signals. And so in the information bottleneck, typically you have data from some input signals that you know and some output that you know. So this thing is given, this key, I don't know, output given signals or and yeah, so the joint distribution between output and signals is given. And so it's interesting actually, because I think a lot of people yesterday have talked about sort of these architectures where maybe you have like now some small thing here and there's some compression and then it sort of opens up again. My understanding of the information bottleneck is that it won't do this opening up by definition of how you're choosing this intermediate variable. But that what it actually will do is basically if you have sort of your large signal space and then the space in between, you can think of, you already have maybe some sort of information funnel from signal to output. And then what C does is that it finds the best possible compression of the signal space so that you maintain as much information about the output. So essentially, maybe I can draw this more like this almost, right? So it finds the best possible compression, and that's not a trivial task, especially if your signal space is huge, because you could surely find signals that are very uninformative about that. Exactly. But it doesn't, I think it doesn't do this. And I think that's an interesting question, how you do this. It doesn't do this because you're essentially writing down these probability distributions like Markov chains. And so it can't because of the, yeah. Mm-hmm.
Marianne?
Yes.
A question about the data.
Yeah.
How does a typical data set look like? Do you get a time series, and what exactly do you get? And
yeah, I think, so I would say it really, really varies. So with the transcription factors and the fly embryo, I think now you're getting time series of inputs as well. But in the fly embryo, you're a little bit limited by essentially these developmental timescales. So maybe what I can quickly say, so that you start, of course, with one nucleus, and then eventually you get to sort of 14 cell cycles of divisions, and these cell cycles have a specific time. So you only ever have the time of the nuclear cell cycle available. Here, you definitely get time series. So here I would say in the mouse, you do often get time series, even though people, of course, also do fixed experiments. But also, I would say with heavy noise.
that is going into the system.
Yes. So, sorry. Yes. So it's basically you would basically maybe have something that mimics notch. There are these Hess genes, for example, and then in experiments they would track these Hess genes for a particular cell as a function of time, but then not for all cells. So that's, I think, at least in the experiments that I've worked with, you either get sort of fixed data from a lot of cells, or you get individual cells that are labeled that sort of move around for a certain amount of time until they move out of view. And then during that motion for individual cells, you get essentially like individual time points where you have inputs. But I would say most of the data that I've worked with is still stationary. And in that case, even though we know it's a high dimensional space, it's actually surprising how much of it people focus on one input. So that's also the data that I've worked with. With the gap genes, of course, you also have multiple inputs. So then it's I would say a constant time, but multiple input data set. And there is, maybe just to complete this, at least with some of the experiments that I've worked with, I think the noise is really non-negligible also on the input side.
And the output would be the fate of the cell.
Yes, so on the output then people either have sort of a clear assumption, like in the fly embryo, they just say, well, we know that the fly embryo, that there are these segments. We know that these segments later on, essentially, that these segments all have distinct nuclear identities. So we can essentially label these segments by some axis parameter. Or what you can do is basically, so this is also what people often do, is that they basically plot these different marker as an output on some axis, and then they say, okay, how much brachyury do I have versus how much SOX do I have? Where am I in this sort of output field? And that, I think with most of the data that I've worked with, it's been a constant time, so single time point at some end. But I think people also do time varying. Yeah.
Thank you.
Mm-hmm. And here in the middle, essentially, you can, of course, basically it depends on what technique you're doing, MS2 or FISH or something. But there you can also check, on maybe both on the, especially on the mRNA label. I think that's the most dominant data. What type of mRNAs are you making? Or you can, of course, sequence and sort of then get also fixed time points in the middle. And this is basically as a function of, you can get this as a function of time. Yeah, okay. Yes. So, okay. So now what I wanted to do is to essentially sketch out one calculation just so that people maybe who haven't seen a lot of these information theory approaches before maybe get a little bit of a feeling of how one would do this type of calculation. And so I'm going to choose this because it's a simple one. It's among other things in Bill's books, but it's also in many books. So what you typically do when you have, and I'm going to do this for two variables, so an input and an output, and I'm going to assume that they are both one-dimensional because that's the thing where you can do this the most easily. So what you're essentially doing is that you have sort of an input and an output, and you now want to optimize this probability distribution of the input to maximize the information between the input and the output. So what you basically could think of is that you have a signal input and an output, and now you're saying my map is fixed. So I know to some extent, given the signal, how much of this particular gene or something is my cell going to produce and with what noise. So you could, for example, imagine that there's like a linear map or some sort of bent map, and then there's some sort of noise around this map, and those things you know. And it could also be like some sort of gamma distribution, of course. But you assume that this thing is known. And the limit where we can do this calculation very easily is essentially in the limit of small noise. And so small noise, because then your type of distribution barely matters, and you can essentially assume it's Gaussian. And what's also important is that this mean function O given S, that is monotonic, because if it's non-monotonic, you get into all sorts of trouble. Okay. And so in that case, how you can do this optimization is that you basically write down your mutual information, which can be expressed in terms of an entropy. And I'll say this in a second more clearly. In terms of an entropy and a conditional entropy. For those of you who haven't seen this, in this case, the entropy is the entropy in this variable x would be this. And so you can essentially, what you can do is essentially you can rewrite this thing in this information here like so, where you basically just rewrote this in terms of the conditional entropy, and then you can pull those two terms apart and you get one term that looks like this, so that's the entropy, and one term that looks like, that has this conditional guy in here, so that's the conditional entropy. So you can do that. Okay, and so now what you can also do is to basically, so we can write this in both ways. It's a symmetric thing, and it doesn't matter which way we write it. So what you can now do is that you essentially say, well, okay, so I know this guy. I know HO given S because I assumed that I'm in the small noise limit. So I can essentially assume that this map between O and S is Gaussian. And for Gaussian variables, you can calculate the information. Let me not worry about these twos. And you can get this expression where you do the average over S. So I haven't shown you how to do this, but what you can essentially, the way you see this is that basically you have an integral ds do, do given S, ds log do given S. And now what you can do is that you can insert this guy to be a Gaussian. So you have something that's e to the something something, and then you have the same term here. So that integral you can do if you pull out this guy in front and say, okay, this is slowly varying, so I don't need to worry about this for now. And then you can do this integral and obtain essentially this. So basically I'm saying if your probability distributions are Gaussian, you should know what the conditional entropies are and you can calculate this. And now you do one trick, which you can either do on this level or on this level. So I'm going to do it on this level, which is that you basically say in the small noise approximation, what I can do is that I can say that my noise in O, over this noise in S, which I can, which I would use if I use the probability distribution the other way around, that's just do ds. So this is then like do ds. If you wanted to start here, you could say, well, I know that for a simple probability distributions, you can write, you can essentially just do a variable transform like this, and then you get, and then you get similarly that this is this. So it doesn't matter how you start, maybe to not confuse everyone with this. So essentially what you can do, and one thing that I'm not going to talk about is that we always use the absolute value here, which is actually interesting because these sort of derivatives were sort of mentioned yesterday in Suri's talk. But so we have sort of this absolute value for these derivatives, and then you can basically say that this this error here, if you now want to use this guy, so if I want to use HS given O, that just depends on this noise, which I can now write as, so that would have been this noise in S, and I can now write this as essentially this noise in O times do ds to the -1, -2. Okay. Are people still, I think I see some nodding faces, so that's still sort of clear. I think all we've done is we've assumed these things are Gaussian, and then we've played around with the noise. And so now that you have this expression, you can basically maximize your information over PS by just writing down the individual terms. So you want to basically have some integral ds log PS minus some integral ds log, and then some constant. Let me pull out the constants. Times some constants. Okay, and now you can do these partial, derivatives, and what you get when you essentially now take the ISO with respect to PS, you see that you basically get, from this term, you get something that's a constant, like a 1, but you also get a constant from this term, so I'll just write some sort of constant down. Then you get, so a constant because you basically, this log gives you 1 over PS, which cancels this PS. Then you get this log term, which is this log PS. And then from the second term, you get also a minus log noise dO ds to the -1. Okay, and so now if I've not made terrible mistakes, we should get essentially that this log es sigma o dO ds is a constant, and that therefore es can be written approximately as 1 over this sigma o dO ds. That looks approximately right, just because basically, maybe you can think of it this way. Basically, if you had a map between O and S, and like one thing that you could typically say is that maybe this map is linear, but maybe your noise increases, right? So then just by intuition, what you would want to do is that you would want to use, if you want to maximize signals through a noisy map like this, you would want to use more signals in this area where the noise is small. You know, because then basically if you have, I mean, I don't know, if you write, I think the example people always use is the example of the noisy typewriter. If you write with a typewriter and your letters occasionally get confused, you know, assume some letters get confused less than others, you would use, of course, more of the letters that get less confused than the ones of the other ones, which get all smeared out. So that's what this is saying, essentially, to some extent too, that your probability distribution should be higher where the noise is small. Okay. And so now, of course, I said, you know, these approaches are model-free, and that is true. But of course, the hope, at least for me, would always be to maybe find some type of approaches to the models also from this InfoMax approaches. And what you can maybe see here is that you have this, you know, you have a function here of O and S. So you could, of course, now also say, given if my noise is fixed and my PS is fixed, how do I optimize this particular function? Okay. So this was an example. This was meant to be an example for these InfoMax approaches. Oh, yes. Yes. Sorry? No, sorry. In this case, we were maximizing the information between S and O. So we were maximizing essentially how good of a message can I send? You know, assume I have noisy signals and some outputs, how good of a message can we send? But we had assumed maybe like a little bit like in gene regulation that this map between the signal and the output is fixed. So we were optimizing over the signals. So we were saying what type of signals, or like how many, you know, how often should my organism see a signal of a particular concentration value versus another concentration value, if you think that the information is in the concentrations. We are maximizing the information and over the signal distribution. Over the signal distribution, exactly, yeah, thank you. Mm-hmm. Yes. So, I mean, so basically, I think when it's small noise, I would say, people would say no, but I would say essentially that's what you're doing, right? Because, I mean, when you have small noise, you're essentially just saying that higher order noise terms won't come in. So I think it's equivalent, yeah. I think that they're not separate or something, yeah. Mm-hmm. Okay, yes, and I should say that what I've shown you is essentially for small noise where these things can be done analytically. When the noise gets larger, for this type of approach, there are methods out there like the Plahu-Darimoto method, and I'll maybe mention some of them in my talk, but yeah, so there are numerical methods that can help you do this when you don't have the small noise, which is really where I feel like the only limit, apart from this huge noise limit, where we can do something analytically. Okay, so the purpose of this was essentially to give people a feeling of how some of these optimizations work. But what I now wanted to do is to sort of go back to this idea that I think what both approaches, both the model approach as well as the optimization approach have in common is essentially that they are kind of considering the landscape that you're looking at. So, and I think what's interesting, because we're talking about this high-dimensional biology, I think what we've heard in a few talks already is that even though we have a lot of high dimensions, often there's a latent space that's a little bit lower dimensional. And I think that's what I wanted to get to, because I think one attractive view of seeing this lower dimensionality comes from this idea or this work from Jim Sethna and others that essentially thinks about these sloppy models or sloppiness in general. So I wanted to explain maybe briefly what they are and how they kind of affect the landscape, both in the model view as well as in the optimization view. So maybe just write this down correctly. So I think what Jim Sethna, the reference that I'll use is this one. Actually, maybe I should ask, because I think probably people do know this, but I still wanted to sketch it out, but maybe that gives me an idea of how fast to sketch it out. Who knows or like is super comfortable with the sloppy models? Ah, maybe I should have not said super. Okay, maybe I'll, but then I'll sketch it out. Anyway, so basically what Jim Sethna and others were sort of doing was that they had models. In this case, you know, they had some parameters Y for a lot of different biochemical processes. And they looked in various papers how, you know, for basically standard models that people had done for these variables Y. So in this case, my output is essentially Y as a function of T, it does something. And for these variables Y as a function of the parameters that people had measured or suggested in various papers for these models. So what they then did is that they basically wrote down what I would argue is to some extent a cost function, which is essentially an error for how this Y depended on these parameters. So what they did is that they wrote down, you know, what does Y look like when I have a different parameter versus, sorry, and these things are vectors, I should say. What does Y look like when I have a different set of parameters minus what does Y look like when I use the set of parameters that the people had suggested or that people had measured in these models. Let me assume that I normalize that correctly. And now because Y is a time series, I also need to sort of integrate over time to make sure that I take into account all these different time points. And then there are some sort of additional sums out here for these number of these parameters. So it's essentially like an error in terms of how Y, my model variable, diverges from the Y that I would have if I had the right parameters. Yes, and so then what they did is that basically they used this error cost function and tried to calculate a Hessian of this cost function with respect to the parameters, which in this case they did with the log of the parameters, so log theta I d log theta k. Here, yes, sorry, yes. Thank you. Perfect. Okay, so they calculated these Hessians and then they looked at the eigenvalues of this Hessian. And they looked at the spectrum of these eigenvalues. And what they found, like if we denote these eigenvalues by lambda, there are sort of these beautiful plots in the paper where essentially this parameter has like, you know, different models for different, in this case, developmental, but also metabolic processes. Here you plot this Y and you plot Y on a log scale. And then you find these like sort of very crazy sets of eigenvalues that span essentially multiple decades and often actually sort of surprisingly semi-regularly. So the idea is that basically some of these eigenvalues of this Hessian, some of these eigenvalues, which basically corresponds to eigenvectors in this model space. So some eigendirections in this model space matter essentially nothing at all towards getting for you to get the right model, for you to get the right Y, whereas other parameters are going to matter hugely. And so I think this idea is sort of somewhat attractive because it says essentially that, you know, and we've heard this a lot with the latent spaces, that I guess some parameters matter much more than other. And I don't know if the right way to say is that you can essentially think of an effectively much more low dimensional system, but to some extent, it's definitely not, you know, all these dimensions, even though even these dimensions, of course, matter weakly. Okay, so that was to some extent the sloppy approaches in the model. So we can look at variations in model parameters and find that that not all parameters matter equally. Yeah, some maybe more important than others. Okay, I don't maybe need to write this down. Okay, and so now the idea is that basically we can do this approach also, of course, when you have an optimization function, when you have a landscape that you optimize from. So in this optimization view, you typically write down some cost function f, and f can now be, you know, this mutual information that we were talking about, and so on. And you know, in this case, the mutual information in the example that I've done, you know, we've optimized over PS. But I could now also make PS, for example, one thing that I could do, which I didn't do, was that basically when you look at this example that I've just done for high noise, you will find that PS actually becomes discrete. So far, you know, I've basically plotted PS over S. And in the example that we had, it should be some sort of decaying function. But in the high noise limit, what you get is basically that you pick individual S's. You pick this S and this S and this S and this S. Sorry? Sorry. Okay. Okay. Thank you. So, I was going to say in the high noise limit, I said what I wanted to do is to basically say, you know, why are there things that I can vary? So of course here I can vary PS, but S is a function. It's always more easy to vary something discrete. So I wanted to just, so people understand what I'm going to vary or what one can vary, I wanted to say this PS can also be discrete if my noise in this map between O and S is high. So before I had this like small noise limit. If the noise is huge, you can again sort of think of this noisy typewriter. If the noise is huge, you know, I might just use letter A and letter Z because I know they are furthest away in my alphabet, so I have the least chance to confuse them. And in that case, my PS would be a prior that has essentially 2 delta functions. And so then these things I could also parameterize and say, now let me vary the position of these delta functions. So I'm just saying that, you know, because here we had these parameters, and in information, of course, I can either do this or I can sort of include other parameters with respect to which I now want to vary this cost function, this optimization goal. So now what I can do is essentially that I basically have a probability distribution of parameters that determines my cost function. And I can say that basically at an optimum, some sort of optimum here, I can say that at an optimum, I can make an expansion of this F to second order. So I can essentially say that F is my F max plus some sum over ij, like this Hessian, theta I minus theta I star, or at the optimum, and theta j minus theta j bar at the optimum, where these parameters now denote my optimum. Okay, so far I've not done very much differently. I'm just saying I can, you know, like before, I have a cost function. I can essentially expand my cost function. I'm now going to look at the Hessian with respect of this cost function with respect to the parameter. And of course, it shouldn't surprise much that you actually find the same. You find that the eigenvalues of these Hessians are also spaced sloppily. The eigenvalues lambda also have the sloppy eigenvalue spacing, which essentially means that basically lambda mu goes as some lambda max e to the minus alpha mu. So you have some maximum lambda, and then everything else decays exponentially from there. And so now the thing that one can actually do is to basically say what is attractive about this are sort of 2 things. So first of all, it means that these optimum in biology are no longer individual points. So you can have a large region of optima that are extremely close in terms of the value of this cost function to the real optimum, but the parameter space can vary hugely, right? So this sort of maybe old notion that basically an optimum means, you know, this system is the best at these specific values of the parameters, that no longer holds because you can actually really vary these parameters significantly. And then the other thing, and yeah, I should say this is a paper that we have on the archive with Bill and lots of other people. So Bill Bialik, Chase Goddard, Caroline Holmes, Kamesh Krishnamurti, Rich Pang, Stephanie Palmer, David Schwab, Sussman. So it's a lot of people that I always forget the names. I mean, I do know the names, but yeah, so it's on the archive, like I think in May. And so the other argument that I think we are trying to make in this paper is that basically if you do a maximum end optimization, so if you basically assume that you are the one who has to pick these parameters, if you now have to pick these parameters based on some maximum entropy optimization where you have essentially this behavior with respect to your parameters, then what you can find is essentially that the difference of this mean of this cost function and your maximum value become exponentially close, because essentially, so let me make this more clear. So let me say I want to now calculate a mean cost function, which I can of course do. That's just integrating over my parameters theta, e theta, f of theta. And now I want to essentially calculate, how much is this mean away from this maximum of my cost function. So I want to calculate some sort of delta f. And since the maximum is higher, probably I should have written it maybe more like this, so that I have a positive number. So I can calculate this delta f. And I can find that this delta f essentially becomes 0 as the dimensions go to infinity, because the assumption that we are essentially making is that as the dimension goes to infinity, we will have this type of scaling in the parameters. With this type of scaling in the parameters, you can calculate the entropy as a function of t. So we can calculate the entropy, which is just this thing. I can then insert my maximum entropy expression into here. I can calculate the entropy, and then overall, I can essentially find for this delta f that it goes to 0 as the dimensions increase, which I think kind of makes sense, because it's just saying that, I think the new point is basically saying that a maximum entropy approach will get you close to an optimum because the landscape is sloppy. And I think the reason, again, why it's attractive is because it sort of says, an optimum, many things are optimal, but of course not trying to find the optimum is not good. So in the sense, like trying to find the optimum with this maximum end distribution is the thing that you should do to get close to the optimum. Okay, so that's what I wanted to sort of say about this, like maybe unifying to some extent. I don't know, it's a big word, but I just wanted to say that both in the model space as well as in the optimization space, you can use a different language, the same language, to think about these high dimensions. And I think the sloppiness, or I think, yeah, Arvind and Jörn Dunkel use maybe more soft modes for this type of approach. I think that can go in the right direction. And I think one maybe criticism that you could say of the sloppiness is, what if you have a multidimensional landscape that has a ton of separate optima? But I think to that, I think that is true, and that is a fair point. But I think there's work both from the development community. So I think Marianne Bauer has a paper where they essentially say in signaling pathways, it seems, funnily enough, that some of these optima are aligned in some space, so that you could actually maybe to first approximation, this is my interpretation, not theirs, but you could maybe to first interpretation say that there's like, again, one sloppy directions because you have lots of different optima aligned in a sloppy direction. So this was mentioned in an eLife from, I forget the year, recent. And then there's also work from, on the machine learning side from the Salmhofer group, which I think, that's longer ago, which I think says that basically in these machine learning landscapes, often the minima are actually connected. So, or at least you can find some basis function that you don't have something like this, but that you have like some smooth curved space. And of course, with the circular, our sloppiness doesn't help much. But I think if it's like a smooth curved space, you could again, calculate eigenvalues along these directions, and you would find some sloppy approaches. So I think that these things are maybe meaningful is maybe a fair thing to say. Okay. Yeah, so maybe I should actually ask if there are questions. Yes. So we are expanding. We are expanding the cost. We are basically saying the mutual information is one example for a cost function. Yes. Interesting. You're saying, is it like a Fisher type thing? So basically, in the paper, we've used a different type, like a large set of different cost functions. And so I guess, yeah, I'm not sure. Is it? So it's actually an interesting question. I don't know. I guess maybe one could think of it as like Fisher thing, but I know that for some other parts with the machine learning stuff, they've actually used the Fisher information because it was easier to work with. Yeah. Mm-hmm. Yes, that's right. So we have, so basically we've often assumed this form, which we found, but you can essentially, as long as your lambdas decay sharp enough, you can always essentially plug this equation into here, express S in terms of lambda and T, and then because you can essentially, for this guy, you can use equipartition, so you know that it's basically D over 2T. You use the T that you find from here in there, and as long as the lambdas are decaying, the dimensionality will pull that down. If the lambdas are all the same, you don't get it. Mm-hmm. Okay, so I think maybe what I'll do very quickly, just because it's purely for selfish reasons, I'll actually, Sid, what do you think? I wanted to maybe very briefly talk a little bit about the info bottleneck. But it's, yeah, so I had thought I would have a little more, but that's okay. So I'll maybe just, I'll make sure I'll end on time. I just wanted to talk about the information bottleneck because I thought, as I was saying before, like I think it's a really nice, strong framework, but I think it would be kind of nice to also maybe sort of, yeah, I think there are some people who are really big fans of the info bottleneck and some people who are not, and I think it would be nice to maybe have a more closer discussion in terms of what is good about it and what, you know, potentially could be improved. So the information bottleneck approach is to basically do a similar optimization to what I was sketching out before. As Kabir had asked, what you're essentially doing in the information bottleneck is to basically have your signal space, you find a compression C of the signal space, and then based on this compression, you essentially try to infer an output. And the reason why you're doing this is because you have this implicit assumption that basically the processing layer that needs to process the signals needs to do so efficiently. So it needs to essentially look at parts of the signals that are most informative. You could now, of course, argue that this processing layer can also generate information, for example, because of nonlinear dynamics approaches, out of equilibrium thermodynamics approaches, or you know, mutual repressive patterns. So you could say that the information flow, which in the information bottleneck is sort of always limited by the data that you have. So you have this information between S and O. That there's some sort of structure like this. And the reason why I think that is important in the information bottleneck is because people typically plot things on what's called the information plane. So this information plane is basically that you have the information between C and the outputs on the y-axis. So this is the thing you want to maximize. This is the thing you want to have as large as possible. And you have the information between C and S on the x-axis. So this is the thing that where you're measuring with CS. And this is the thing where you want to, in principle, be as noisy as possible because you want to find the smallest possible like compression that can map all your signals but still give you the correct output. But the argument that I want, or the thing that I wanted to clarify or to sort of say is that, you know, we now all know that this information between C and the output in this setup is always limited by the information between S and O. So that's the thing that I have given, and I want to infer the information in C here. And the other point is that it's also limited by the diagonal. So basically the reason why that is because when you have a particular, when you make an approximation for PC given S, any type of map that you start out with, then what you do is that you calculate the map between C and O by essentially integrating out this S dependence. So you're essentially saying that this is a Markov chain through S. This is the thing that I know, PS given O I know, and now I have a guess for PC given S, and I'm integrating over S. And sorry, and I should say this was, of course, when Naftali Tishby and Bill and others introduced this the information bottlenecks. So this is a paper by them. They did this, I think, fully on purpose, because they said, this is the best possible thing I can do, given the fact that I don't know what this PC given O is. And so the example that I think one can use is that, S is some signal, or S is maybe, some sort of image of some animal. And then here on the O side, you maybe have this label. And so if you now have any type of compression, I don't know, some sort of thing, what you would do is that you basically compare it to your state of signals, and then you say, okay, well, if it matches this signal best, then I'm going to go to cats. So this is how you find this map. So they did this on purpose, but I think it does limit, it definitely limits where your optimal solution can be on the information plane, because it's going to always be in this part of this information plane. And so what you do, maybe just to end is that when you find this optimal compression, what you typically get is a line from data. So you get some sort of line. This is the numerical solution this is the numerical solution of this optimal compression for different ICS. So basically, for every value of ICS, you get an optimal PC given S. That optimal PC given S then gives you an optimal ICO. And so given that you basically don't know how noisy your ICS should be, you get lots of different PCSs on a line, and practically you constrain the value of this variable with this Lagrangian multiplier lambda. And so the reason why I'm saying this is because sort of in this phase diagram, you're now looking for the optimal compression, and often this optimal compression can actually be pretty far away from these two lines, which are essentially the data processing inequality. So in neuronal systems, often you're actually quite far away. And so that's why people come up with other approaches, which is basically trying to calculate what's called a strong data processing inequality, which is more data specific. And that slope of that is going to give you things that are practically possible. So I would say here, above the data processing inequality, you're in a completely impossible regime of the phase diagram. And I've now sort of started to label this because I've noticed that Lenka does it. And I think it does make it clear. So in this phase diagram, there are regions that are totally impossible. Then this is the data. And I would say getting close to the data here is hard. Here is practically impossible because you can never do better as the data. And the part of the machine learning research community that is interested in these strong data processing inequalities essentially tries to find a larger phase space that's practically impossible. Because if from my data, a lot of the time, the strong data processing and the data processing inequality are essentially the same. But if, of course, you have a strong data processing inequality that's further down, your data is going to be here. So they are essentially trying to put these different regions of the phase space to sort of classifying more easily what's accessible. And then somewhere here, I guess it's easy, at least for my part. Okay, I think that's all I'm going to get round to. Yeah, maybe if there is. Yes. Aha. Yes. Sorry? So that is yes. So, okay. So that is the information between this compressed layer and the output. So that basically tells me, if you have molecular signals and then you have all these promoters in here that process these signals, how much do I know on this level? How much can I, once the promoters are starting to express the genes, how much can they know about the future cell fates? So that's the information about for this compressed layer between the output. And what we're trying to do with the information bottleneck is to essentially find this compressed layer or find a map from the signal into this compressed layer. Yes. Sorry, C and S. Yeah, I should have. So it's I, C, and S. So that's the information between the signal and C. So that is how noisy does my intermediate layer process the signal. And in principle, where you want to ideally be is you want to basically have the noisiest possible processing of your signal and the best possible output. Because then, so you have to think about maybe this intermediate processing layer has to measure, right? It has to measure these transcription factors. It has to make new genes. And so I think the concept idea in this information bottleneck applied to molecular sensing is that there is essentially a constraint on how much this intermediate layer can do. Because you want to maybe not measure transcription factors when you kind of already know what you need to do. Or you want to maybe not make more molecules when the couple of molecules that you've made are already enough for you to determine your sulfates. So in principle, you want to be as low on this axis as possible. But because you don't know yet essentially what your best possible compression is, you need to essentially first calculate the best possible compression, and then you can say, maybe heuristically, where on this thing should I now be to optimally get information?
Maybe one last question. Yeah.
Right. That straight line essentially says that once you compress the information, you can't extract more out of it. In
this one?
The
diagonal?
Diagonal. Yeah.
Yes, the diagonal essentially says that, yes, you could see it like that, that basically once you compress the information, you can't get more than you would get from this comparison, essentially.
But you made a comment on the non-equilibrium nature of...
So this is a little bit my strong hunch. I think that is correct, but I haven't sort of calculated anything. But I think if you basically say, or like basically what Sean was doing yesterday was that he basically infers some autoencoder, where then I think he also optimized some network here, right? So this map on this side. We, in the information bottleneck, we don't necessarily optimize this map on this side. We just calculate it through there. And so basically, if you, I don't know, either optimize this, or if you assume that they're sort of out of equilibrium, rates, I think then my strong hunch would be that then you don't have this problem.
So initially when you draw that picture of the one with the bottleneck,
yes,
and the one where you just truncate and the processing just tries to modulate the compression. You had these
yeah, you can see it like that. I think the optimal processing essentially tries to modulate the best possible compression of the signal to the output. Yes, but it does not generate information on this side.
So the diagonal would be aligned with the second picture that you drew? The fact that you have a diagonal?
Yes, I think that's right. I think the fact that I have a diagonal is in line with this picture.
Right.
Yes. Okay. Okay. Cool.